Hello and welcome. Dear students, today we are going to discuss an important topic, that is holoenzymes, apoenzymes, cofactors, coenzyme, prosthetic groups, metalloenzymes, monomeric and oligomeric enzymes. The main objectives of today's lecture are to understand the concept of holoenzyme, an overview of apoenzyme, to understand the concept of coenzymes and their classification, to get a general idea about cofactor and its types, monomeric and oligomeric enzymes. Let's first of all give a brief summary about today's topic. An apoenzyme with its cofactor is known as holoenzyme. It's catalytically active and complete. Many cofactors are not covalently bound, but as an alternative are tightly bound. However, organic prosthetic groups such as an iron or a vitamin can be covalently bound. DNA polymerase and RNA polymerase are examples of holoenzymes. These contain multiple protein subunits. The enzyme before binding with the cofactor is known as apoenzyme, or we can define apoenzyme as the protein part of the enzyme which lacks the cofactor. Apoenzyme is catalytically inactive and incomplete. It forms an active enzyme system upon combining with the coenzyme and determines the specificity of the system for a substrate. Coenzymes are defined as small non-peptide compounds which are organic in nature and essential for enzymatic reactions. They are considered as a special class of substrates because their nature is changed in an enzyme catalyzed reaction. They also work as second substrates with many enzymes. For example, NADH is used as a coenzyme in more than 700 enzyme catalyzed reactions. The chemically changed coenzymes are regenerated at a continuous level and their concentrations are maintained at intracellular level. For example, NAD positive is reduced in glycolysis to NADH, that is oxidized back to NAD positive in mitochondria by respiratory chain reaction. Whereas cofactor is a non-protein substance that forms complex with some enzymes and assists them in biological reactions. Cofactors may consist of organic compounds, metal ions, or some other substance that have useful properties usually not found in amino acids. Some cofactors can be made inside the body, example ATP, while others are obtained in food. At the biochemical level, cofactors are essential in understanding how biological reactions proceed. The presence or absence of cofactors in a biological reaction may decide how quickly reaction will proceed. A prosthetic group is tightly bound, specific non-polypeptide unit required for the biological function of some proteins. The prosthetic group may be organic, such as vitamin, sugar or lipid, or inorganic, such as the metal ion, but is not composed of amino acids. Prosthetic groups are bound tightly to proteins and may even be attached through a covalent bond as opposed to co-substrates which are loosely bound. Whereas metalloenzymes are those metalloproteins that have one labile coordination site around the metal center. The shape of the active site is crucial in enzyme activity. The metal ion is positioned in a pocket whose shape fits the substrate. Monomeric enzymes consist of one polypeptide chain. These enzymes generally help in digestive processes and are functional in their tertiary forms, whereas oligomeric enzymes consist of more than one polypeptide chain. The molecular weight of oligomeric enzymes is usually more than 35,000 kilodaltons. They are functional in their quaternary structure. Now, let us talk about the first objective of today's lecture, that is holoenzyme. An apoenzyme with its cofactor is called as holoenzyme. It's catalytically active and complete. Many cofactors are not covalently bound, but as an alternative are tightly bound. However, organic prosthetic groups such as an iron ion or a vitamin can be covalently bound. DNA polymerase and RNA polymerase are examples of holoenzymes. These contain multiple protein subunits. The complete complexes contain all the subunits necessary for activity. DNA polymerase is an example of holoenzyme. It catalyzes the polymerization of deoxyribonucleotides into DNA strand. DNA polymerase is an active participant in DNA replication. The intact DNA is read by it, and this DNA strand acts as a template which is used to synthesize the new strand. The newly polymerized DNA strand is complementary to the template strand and identical to template's original partner strand. DNA polymerase uses a magnesium ion for catalytic activity. RNA polymerase is also a holoenzyme that catalyzes RNA. RNA polymerase is needed for constructing RNA chains from DNA genes as templates, a process known as transcription. It polymerizes ribonucleotides at the 3' end of an RNA transcript. Now, coming on to the next objective, that's apoenzyme. The enzyme before binding with the cofactor is known as apoenzyme, or we can define apoenzyme as the protein part of the enzyme which lacks the cofactor. Apoenzyme is catalytically inactive and incomplete. It forms an active enzyme system upon combining with the coenzyme and determines the specificity of the system for the substrate. There are a number of cofactors that bind with apoenzymes to form holoenzymes. Common coenzymes that bind with apoenzymes are NAD+, FAD, coenzyme A, vitamin B and vitamin C, iron, copper, calcium, magnesium, zinc, etc. are common metal ions that bind with apoenzyme to form holoenzymes. Cofactors bind either loosely or tightly with the apoenzyme to form holoenzyme. Apoenzyme is again formed once the cofactor is removed from the holoenzyme. The presence of the cofactor at the active site of the apoenzyme is essential because they provide groups or sites that the protein part of the enzyme does not possess to catalyze the reaction. Now, let us discuss another objective, that's coenzyme and their classification. Coenzymes are defined as small non-peptide compounds which are organic in nature and essential for enzymatic reactions. They are considered as a special class of substrates because their nature is changed in an enzyme catalyzed reaction. They also work as second substrates with many enzymes. For example, NADH is used as a coenzyme in more than 700 enzyme catalyzed reactions. The chemically changed coenzymes are regenerated at a continuous level and their concentrations are maintained at intracellular level. For example, NAD positive is reduced in glycolysis to NADH, that's oxidized back to NAD positive in mitochondria by respiratory chain reaction. This continuous regeneration means that even small amounts of coenzymes are used very intensively, such as in human brains, which turn around approximately 3.3 kg of NADH each day, while it contains only 75 grams of NADH. Coenzymes are heat stable and act as group transfer reagents which are linked to enzymes by non-covalent forces. They can be separated from the enzymes by dialysis. Now, the classification of coenzymes. Coenzymes are classified according to the reactions they are involved in. Redox coenzymes. Nicotinamide adenine dinucleotide phosphate. The purine nucleotide containing NAD positive and NADP positive are coenzymes from class of dehydrogenases. In addition, they contain redox active nicotinamide, that is vitamin B3, while NADH transfers protons and electrons from catabolic pathways to the respiratory chain NADPH. Reduced form of NAD positive is the most essential proton donor involved in the biosynthesis. Flavin mononucleotide FMN and flavin adenine dinucleotide FAD. The flavin containing FMN and FAD are coenzymes of dehydrogenases, oxidases and monooxygenases. In addition to redox active flavin, they contain phosphorylated ribitol attached to the flavin ring. Riboflavin is known as vitamin B2. FAD is produced from FMN by reaction with AMP, that is adenosine monophosphate. In contrast to NAD, P positive, both FMN and FAD are covalently bound to the enzymes as prosthetic groups. Ubiquinone. Ubiquinone or coenzyme Q transfers electrons in respiratory chain. In addition to quinone ring, it contains isoprenoid chain that holds ubiquinone in inner mitochondrial membrane, where it's freely mobile. Next is L-ascorbic acid. L-ascorbic acid is a coenzyme belonging to the class of monooxygenases and dioxygenases. Most of mammals, except higher primates including man, synthesize L-ascorbic acid from glucose for the hydroxylation of proline and lysine during the synthesis of collagen. The second class of coenzymes. Coenzymes transferring 1 carbon units. Tetrahydrofolic acid comprises of 4 aminobenzoic acid with pteridine ring attached to it via carboxyl group. This coenzyme transfers 1 carbon residue in different oxidation states like oxidation of formic acid, oxidation of methanol, etc. S-adenosyl methionine. S-adenosyl methionine is synthesized from adenosine triphosphate, and methionine is donor of methyl group involved in many methylation reactions like synthesis of epinephrine from norepinephrine or in methylation of DNA. Methylcobalamin. Methylcobalamin, a methyl derivative of cobalamin, is a coenzyme from methyl transferases. It's used in the treatment of peripheral neuropathy and diabetic neuropathy. Biotin. Biotin belongs to the class of carboxylases. In presence of ATP, it reacts with bicarbonate to form N-carboxybiotin, activated form of carbon dioxide that is then transferred to other molecules, example in the form of oxaloacetic acid from pyruvic acid. Vitamin K. Vitamin K comprises of structurally linked compounds that work as coenzymes of gamma-glutamyl carboxylase. Carboxylation of glutamic acid residues to gamma-carboxyglutamic acid that is catalyzed by gamma-glutamyl carboxylase occurs in the proteins involved in blood clotting cascade factors 2nd, 7th, 9th and 10th and some bone proteins. Third is coenzymes transferring 2 and more carbon units. Thiamine diphosphate. Thiamine diphosphate, phosphoderivative of thiamine, vitamin B1, is a coenzyme of enzymes that are able to active aldehydes and ketones as hydroxyalkyl groups and transfer them to other molecules. Example in conversion of pyruvic acid to acetyl coenzyme A. Oxidative decarboxylation of alpha keto acids take place in which thiamine diphosphate is involved. Next is coenzymes transferring other groups. Pyridoxal phosphate. Pyridoxal phosphate, vitamin B6, is the most important coenzyme in amino acid metabolism. It works as the coenzyme in transamination, decarboxylation and dehydration reactions of amino acids. 3-phosphoadenosine 5-phosphosulfate. 3-phosphoadenosine 5-phosphosulfate serves as a coenzyme in sulfotransferase reactions that are essential for sulfitation of proteoglycans and glycoaminoglycans as well as for synthesis of sulfocerebrosides. Now coming on to the next objective, that's cofactor and its types. Cofactor is a non-protein substance that forms complex with some enzymes and assist them in biological reactions. Cofactors may consist of organic compounds, metal ions or some other substances that have useful properties usually not found in amino acids. Some cofactors can be made inside the body, example ATP, while others are obtained in food. At the biochemical level, cofactors are essential in understanding how biological reactions proceed. The presence or absence of cofactor in a biological reaction may decide how quickly reactions will proceed. Examples of cofactors include metal ions like iron and zinc, vitamin B3, that's thiamine. Now let us discuss the types of cofactors. Vitamins. Vitamins are cofactors in the form of organic compounds that are important for many biochemical reactions. These are obtained outside the body as vitamins cannot be made inside the body with the exception of few. Minerals. Minerals are cofactors that are obtained outside the body. These are important for proper functioning of our body cells. The difference is that while vitamins are organic molecules containing carbon which are often made by other living things, minerals are inorganic substances that occur naturally and are often found in rocks and soil. Organic non-vitamin cofactors. Some cofactors are organic substances not classified as enzymes. Some of these may be made inside our own bodies and so not qualified as vitamins. Organic non-vitamin cofactors include ATP, an essential assistant to many biochemical processes which transfers energy to numerous enzymes, transport proteins and more. Now we will talk about prosthetic groups. A prosthetic group is a tightly bound specific non-polypeptide unit required for the biological function of some proteins. The prosthetic group may be organic such as a vitamin, sugar or lipid, or inorganic such as a metal ion, but is not composed of amino acids. Prosthetic groups are bound tightly to proteins and may even be attached through a covalent bond as opposed to co-substrates which are loosely bound. In enzymes, prosthetic groups are often involved in the active site, playing an important role in the functioning of enzymes. Proteins present with the prosthetic group are called conjugated proteins, and they include glycoproteins, lipoproteins, nucleoproteins, chromoproteins, example hemoglobin, metalloproteins, phosphoproteins. Vitamins are another common prosthetic group. This is one of the reasons why vitamins are required in the human diet. Inorganic prosthetic groups, however, are usually transition metal ions such as iron. Now, let us discuss metalloenzymes. Metalloenzymes are those metalloproteins that have one labile coordination site around the metal center. The shape of the active site is crucial in enzyme activity. The metal ion is positioned in a pocket whose shape fits into the substrate. These metal ion catalyzes chemical reactions that are tough to attain in organic chemistry. Carbonic anhydrase is an important enzyme used in important reactions. In all living organisms, carbon dioxide is the key metabolite which exists in equilibrium with bicarbonate. Bicarbonate is less soluble in lipid membrane in comparison to carbon dioxide. Carbon dioxide can move freely in and out of the cell as compared to bicarbonates. Thus, an important balance between the conversion of bicarbonate to carbon dioxide and the vice versa is required. This interconversion is catalyzed by carbonic anhydrases. These are zinc-containing enzymes found in all living organisms and play an important function in assisting the transportation of carbon dioxide and protons in intracellular space. Now, let us discuss the last objective of this module, that is monomeric and oligomeric enzymes. Monomeric enzymes. Monomeric enzyme consists of one polypeptide chain. These enzymes generally help in digestive processes and are functional in their tertiary forms. They are usually synthesized in their inactive form and are activated at a particular temperature. Examples of monomeric enzymes are ribonuclease, lysozyme, hexokinase, etc. Oligomeric enzymes. Oligomeric enzymes consist of more than one polypeptide chain which are linked to each other by non-covalent interactions and not by peptide bonds. The polypeptide chains are termed as subunits and they may be identical, therefore they are sometimes referred as protomers. Dimeric proteins consist of 2 subunits, trimeric consist of 3 subunits and tetrameric consist of 4 subunits. The molecular weight of oligomeric enzymes is usually more than 35,000 kilodaltons. They are functional in their quaternary structure. Large number of enzymes are oligomeric in nature. For example, aldolase consists of 4 chains. Rubisco of Calvin cycle consists of 24 chains and enolase is a dimer. Likewise, enzymes involved in the glycolysis are either dimeric or tetrameric. Vertebrate dehydrogenase is an example of oligomeric enzymes where each subunit has the same function. Likewise, in the mammary gland, lactose synthase is an oligomeric enzyme where a non-functional subunit modifies the behavior of functional subunit. Tryptophan synthase of E. coli is an example of oligomeric enzyme which consists of 2 different functional subunits. The activities of these enzymes are regulated by more precise way of feedback inhibition. This is because of the fact that many oligomeric enzymes exhibit allostery in which different binding sites interact. Dear students, that was all about today's program. Hope you have understood and enjoyed. Thank you and goodbye.